All right, everybody. Let's go ahead and work through looking at the HIV life cycle. We did look at this last time we had class, just briefly, but I'd love to kind of go over it again with you all because we are going to work into a worksheet that talks more specifically about the actual attachment part here. We're going to look at the actual receptors that HIV uses to attach and mediate entry into host cells such as macrophages or T cells. So yeah, if you look over here to the left, what you see is the HIV virus. You see two segments of RNA. Remember, viruses have some type of nucleic acid as their genome. In this case, HIV is an RNA virus, and it uses this special enzyme called reverse transcriptase to actually copy the RNA, reverse transcriptase, into DNA. This is also, you see the capsid here in purple. So that's the protein layer that surrounds the nucleic acid, but this is also an enveloped virus. And so in addition to the nucleic acid and capsid, you see an envelope or lipid bilayer. It's a membrane. It's a double, membrane, just like you would see on a host cell. And so since this is the outermost layer, what you see are the receptor spikes here that they're labeling as glycoprotein. Okay. Now, this begins like any other viral life cycle. The first thing that has to happen is the receptors here on the virus outermost layer, on the envelope in this case, they must bump into and match a complementary molecule on the surface of the host cell. And so that has happened here. They're not really showing the receptors binding, but that has happened. This then triggers fusion of the viral envelope, the viral membrane, with the membrane that is then surrounding the plasma membrane of the host cell. The two membranes fuse, and this actually will allow entry of the capsid and the nucleic acid into the host cell. Once inside the host cell, the capsid will then disintegrate, break down, and then that releases the RNA. The RNA is then actively copied backwards into DNA. This is why we were calling this reverse transcriptase. ACE means it's an enzyme, and this thing is carrying out transcription in reverse. Yes, so what we're doing is copying RNA backwards to DNA. Then this DNA will migrate into the nucleus where it incorporates or integrates itself into the chromosomal DNA of the host cell. At this point, we call it a provirus. This could go undetected and unexpressed. That means no RNA and proteins made. It can remain dormant and quiet like this up to decades for HIV. We know later, after years have passed, something triggers activation of this. All right, and at that point, what happens is RNA is actively made, and the RNA then actively is translated, and we get viral components. We get, of course, also more copies of the RNA. You see multiple copies of RNA now. You see many copies of the little light purple capsid protein. You also see some of the little glycoprotein receptors. Those guys will insert themselves into the membrane. You also see reverse transcriptase copies that have been made. Those enzymes are made. And what will happen is the capsid and the RNA and reverse transcriptase will self-assemble. The actual glycoproteins, the cell will insert them into its membrane. So that what happens is when the capsid and everything else self-assembles, it can migrate towards and push outward onto the plasma membrane, and it can actually exit the cell by what we call budding. All right, this does not lyse the cell. It's kind of like a dripping faucet with viruses dripping out as each one migrates and steals a section of the host membrane. So that's sort of an overview of the reproductive life cycle of HIV. Let's actually go now and watch a more detailed video that animates this process. Before we do, though, I just want to show you, and I think we looked at this already. This is showing you an electron micrograph of HIV capsid entering. So the membranes have fused, or they're fusing, and this HIV is entering. This is also one that's pushing out, and it's about to exit by budding.
Annenberg Media.
In parts of Africa, almost half of the adult population is infected with HIV.
But this is a virus that has exploited very, very successfully the inability of most women to control their own sexuality.
Today, scientists search for a gene that can keep the virus in check and look for ways to block its infectious path.
Why is the existence of two distinct co-receptors important? It turns out that they have tremendous significance for the real-world biology of HIV.
But can HIV be stopped?
We can't predict the success of DNA as a vaccine approach, but I'm an optimist. I believe we will get HIV to succumb.
In the early 1980s, there was no word for HIV or AIDS. Young gay men in San Francisco were dying from rare cancers and pneumonias as doctors grappled with the unknown. Today, AIDS is a global epidemic, with new cases spread primarily through heterosexual contact and IV drug use. But thousands of scientists around the world are searching for answers. They're building on the knowledge of the virus and the immune system to find new ways to treat and prevent the disease. Eric von Mueller is a real estate agent in San Francisco. In 1984, his partner died from a rare form of pneumonia. Soon after, Eric learned he was HIV positive.
I never expected to see the year 1985. I certainly never expected to see the year 2000. To be alive and see the new millennia was something that just was beyond my hope.
Eric is a long-term non-progressor. He's been infected with HIV for almost 20 years, but he's never developed AIDS.
I've never experienced any symptoms. I've never taken any medications.
Why does Eric remain healthy? Could something in his immune system be keeping the virus in check? The human immune system has two lines of defense: specific immunity and non-specific immunity.
The non-specific, which we call innate, picks up a pattern, so it could say it's a bacteria, so I better react to it.
At the University of California at San Francisco, Dr. Jay Levy has dedicated his research to HIV, AIDS, and immunity.
Specific immunity, which comes later, because it's the non-specific that first responds, sort of gives time for the specific to arm itself. That goes in and says, this is a herpes virus, or this is the AIDS virus, or this is pneumococcus bacteria.
Specific immunity has two branches: the humoral response and the cellular response. The macrophage is often the first type of cell to face a foreign intruder.
So what it does is grinds up the microorganism into small little pieces, maybe 8 or 9 amino acids. And then they get expressed on the surface of the cell. And they then educate the specific immune system that comes in and says, oh, I see this particular small peptide. I'm going to respond to it.
Helper T lymphocytes with a surface protein called CD4 coordinate the cellular response. When activated by an antigen, helper T cells secrete chemical substances called cytokines. Cytokines signal other cells, including the CD8 killer T cells, to migrate towards the foreign organism. The CD8 cells differentiate into active killer T cells and memory T cells. Some of the chemical messengers from helper T cells also stimulate the body's humoral response carried out by B cells. B cells differentiate into plasma cells, which secrete antibodies that attack free bacteria and viruses. They also differentiate into memory B cells.
Once they have destroyed that particular organism, there's no more protein around, so they lose interest, and what happens? They self-destruct. You'll save a few of them. There are memory cells. So if that organism comes in again, they can react very quickly.
With a powerful immune system fighting off disease, why can't the human body control HIV? HIV is a sphere-shaped retrovirus containing RNA and the enzyme reverse transcriptase. The surface of the virus is covered with peg-like projections of the protein GP120. When HIV encounters a T cell, the GP molecules bind with the CD4 receptor and co-receptor. The membrane of the virus and the cell fuse, and the viral core is released into the cell. The core opens, and RNA is released. Reverse transcriptase is attached and produces a single strand of DNA, followed by a second complementary strand. The DNA molecule enters the nucleus of the cell and is incorporated into the genome. At first, the immune system mounts a normal attack and the viral load decreases. But as the immune system battles the virus, it's helping the virus survive.
What is the most susceptible cell to HIV? An activated lymphocyte. So when these activated lymphocytes come in to try to fight, the virus enters them. So it has more cells to infect. Takes advantage of the immune system. How have you been?
For 16 years, Levy's been studying long-term non-progressors like Eric to learn the secrets of their survival.
His lymph nodes, he's got very tiny ones, which I like.
Have they ever been
indicate.
swollen?
No, perfectly healthy. This is key because that's the power of the immune system to control this virus, something that was totally unexpected when we first found the virus.
That's right.
Levy's focusing his research on the proteins secreted by the CD8 T cells.
What we found was this cell in HIV infection doesn't necessarily kill the infected cell, it secretes a protein, which we call CD8 antiviral factor, or CAF, that suppresses the virus in the infected cell so it cannot produce anything. So that cell is carrying a silent gene and can function. And because it is active against all HIV strains, we're active against the monkey AIDS virus. It's non-specific. So therefore, it has to be an innate response.
The lucky few, like Eric, have a strong CAF response and maintain it over time. Those that go on to develop AIDS have lost the response.
My situation has remained unchanged. My CD cells, my ratios have remained constant over the years, and that's wonderful. And who knows? Who knows what tomorrow will bring?
And if that media ends up being toxic, we can't use it.
Right.
In Levy's lab and other labs across the country, the race is on to identify the antiviral factor. While some labs say there's more than one factor involved, Levy believes CAF is a single substance.
How are we going to find it? It's 1 in a million proteins produced by CD8 lymphocytes. So we're doing now the modern technology to find a protein. And that's really interesting because we couldn't have done it 10 years ago.
Using the blood of non-progressors and others, CD8 cells are grown in the lab and allowed to produce CAF.
If we wanted to know, for example, if one particular patient's CD8 cells can suppress viral replication, we would culture that patient's CD8 cells with infected CD4s and then measure the viral output using this assay.
We started out with 2,000 different genes, and then down to 500, and down to 200, and now we're down to 44.
Once CAF is isolated, Levy says the goal is to empower the human immune system to control HIV naturally.
can you give CAF to people, or should you find a way in which you can tickle the CD8 cell so it makes its own CAF, which is the one I would expect. So we are trying to bring back that innate immune response to people who are infected.
And if there's something in my immune system that is common among all long-term non-progressors, and that can be discovered and that can be used to fight this virus, then that would be something that would be really quite incredible.
While some infected people, like Eric, stay healthy, other individuals remain HIV negative despite repeated exposure to the virus. Researchers know that in order for HIV to enter a cell, it must bind to the CD4 receptor on the cell surface. But is that all that's required? At the Institute of Allergies and Infectious Diseases, Dr. Edward Berger has been studying HIV for almost 20 years.
Why is the virus able to get into a human cell that has CD4, but not into a mouse cell that has CD4? There are two simple possibilities. One is that the mouse cell, even though it has CD4, maybe it's got something else that's inhibiting the virus from getting in. The other possibility is that maybe there's an additional thing that's required, and the human cell has it and the mouse cell doesn't.
In the early 90s, Berger and colleagues began testing the possibilities. They constructed a hybrid cell by fusing a human cell and a mouse cell, and put the CD4 molecule on the hybrid.
What we found, we and other people working on this, is that the virus did get into the hybrid. So that led to the notion that there had to be another molecule on the human cell, in addition to CD4, that was required for the virus to get in. And we called that a co-receptor.
But how could they identify the gene that coded for the receptor? Using a collection or library of all the genetic material expressed by the human cell, Berger started mixing the library with mouse cells with CD4. Then he added only the envelope protein from the virus and measured the amount of fusion by reporter gene activation assays.
Here you had no cell fusion,
The more co-receptor present on the cells, the more fusion occurred.
and here even more.
Berger now knew that the library contained a gene that coded for the mystery receptor. Further research revealed a single gene that, when inserted in a mouse cell, produced a surface protein that caused the cell with the HIV envelope protein and the mouse cell to fuse.
The only function that we had for it was that it enabled the HIV virus to fuse with a cell that was expressing CD4, so we gave the protein the name fusin.
At the same time, Berger identified fusion, now called CXCR4. Other researchers identified another co-receptor called CCR5.
Why is the existence of two distinct co-receptors important? It turns out that they have tremendous significance for the real-world biology of HIV, for understanding how HIV gets transmitted from one person to another. The viruses that are present during the asymptomatic phase are preferentially using CCR5, and it's only during the transmission to the symptomatic phase that you start isolating viruses that can use CXCR4.
But scientists know there's a mutant CCR5 gene that fails to produce a functioning receptor.
And it turns out that one of the major explanations for why some people are not infected by HIV despite repeated exposure is that they are carrying only the defective CCR5, and so they're lacking the co-receptor that's required for the initial virus that takes hold in the body. The other remarkable thing is that people that have no functional CCR5, the only medical consequence we know about it is that they are resistant to HIV.
Because people can live without CCR5, it's become a target for new drug development.
The really most promising kinds of CCR5-directed therapies would be small molecular weight chemicals that bind to CCR5 in such a way that they prevent the HIV from using it as a co-receptor. And there's a tremendous effort going on in the pharmaceutical industry for that. There's a real hope that this would provide another weapon against HIV.
Before HIV emerged, scientists thought they could control most viruses with traditional vaccines. They had vaccines for smallpox, measles, and polio. Traditional vaccines are generally made by two approaches: killed virus or live attenuated, a weakened virus that replicates enough to cause an immune response. But HIV presents unique challenges. A live vaccine approach is dangerous because HIV could revert to a form that causes disease, and killed virus vaccines have not worked well in preliminary testing.
HIV is kind of the textbook example of all the problems with making a vaccine.
They were stable.
At the University of Pennsylvania, doctors David Weiner and Jean Boyer have been working to develop a vaccine for HIV that would be as safe as a traditional killed vaccine. But HIV's high replication and mutation rate is just one of many hurdles.
Every time the virus goes through a single replication cycle, every time it grows, it changes a little bit. And so within a single individual, there are many different variations of this virus. And then as they spread through a population, there are many different variations. And so we have to come up with a vaccine that can prevent all these different variations. It also integrates, which means the genetic material of HIV actually is inserted into Y chromosomes. And so once infection happens, the only way to get rid of that infection, we believe, is to destroy that cell. And again, that would require killer T cells.
But how could scientists induce the desired immune response safely? One promising approach is to use DNA as an immunogen or vaccine. Genes for antigenic proteins from the HIV envelope and core are engineered into a bacterial plasmid. Other parts of the virus, like the promoter or the genetic regulatory sequences, are excluded because the virus needs them to replicate.
That's very important. We also delete essential functions in something that's going to go into people, such as the protease genes have destruction in them, and the reverse transcriptase genes. And we also rearrange multiple components of the virus to make sure that it can't reshuffle itself into some novel fashion. And we can test these in laboratory assays.
The genetically altered plasmid is mixed in liquid and injected directly into the muscle. DNA vaccines were first tested on mice in the early 90s.
What we reasoned would happen was now the cell that took up this genetic material would become a factory for whatever the viral protein was that we had encoded. And as soon as it did that, the immune system would now see this cell as having a foreign protein, and that would look to the immune system just like a virally infected cell and teach it how to make the cellular immune response as killer T cells. But because the cell would also leak out antigen, then the B cells would see that antigen and be able to make antibodies. And because the leaked antigen would also stimulate helper T cells, you would get basically all three components of an immune response: the antibodies, the killer T cells, and the helper T cells that are the essence of a live attenuated vaccine without any of the risks of spreading infection.
But would DNA vaccines work?
We published in larger primates that we'd actually prevent HIV infection. And in a smaller primate model, we've shown that we can impact on viral replication, lowering viral replication and protecting CD4 T cells after a viral challenge with a pathogenic virus.
In 1999, scientists began testing DNA vaccines in people. James Burns is HIV positive and undergoing antiviral therapy. He volunteered in a double-blind study testing whether the vaccine would help keep his viral load in check.
The only things I really noticed was like swelling the first day or so, then a little bit of redness. Hi, Doc. Good to see you.
Dr. Rob Roy McGregor administered the first vaccines.
It had been done so much in preclinical studies that, again, I would have been willing to volunteer. So I think a good general principle, if you're doing clinical research, that you have to think it's safe enough that you'd do it.
So far, DNA vaccines have worked poorly in people, but current studies continue to show promise.
These are the animals that were vaccinated and then challenged with our SIV. And you can see in this row and in this row, those are the animals that are controlling viral replication. So we've been moving forward with each primate study we've done, we've gotten better and better responses.
Weiner and Boyer are now investigating DNA vaccines augmented with cytokines like interleukin 12 and 15 that will help boost T-cell responses.
We really want strong T-cell responses. We've included the instructions for expanding T-cell immunity and shown that these work in our animal models, and now we're ready to test them in people. And we think that this will be a magnitude jump, a tenfold enhancement in the vaccine potency.
Sometimes the imagination of a child can help illustrate the goals of research. When Weiner's daughter was young, she drew a picture of her dad's vaccine.
We asked her what was that vaccine about. She says the colors are all the good things you need and none of the bad things that would hurt you. And I think that that's really what a DNA vaccine ultimately could be like. We can take out the things we don't want and put in only what we need and make them function very specifically and in a relatively, hopefully cheap and easily reproducible fashion.
Once thought a radical approach to treating disease, DNA vaccines are rapidly moving into a wide variety of clinical trials. Scientists say that even if the vaccine was only 50% effective and lowered viral loads, it could have a significant impact on transmission rates. But while new treatments spur optimism and hope, many parts of the world remain in the grip of a tragic epidemic. 65 million people in the world are infected with HIV, 25 million in sub-Saharan Africa alone. Most have no hope of getting treatment because the drugs are too expensive and the villages and towns too remote. AIDS is killing the adults who grow the food and earn the money, leaving families with no means of support and millions of children without parents. Laurie Garrett is a Pulitzer Prize-winning medical and science writer. She's covered the AIDS epidemic since the early 1980s.
We can already see that AIDS is going to be the number one destabilizing factor for Africa for years and years to come. And it already has dramatically hurt the economies of the hardest hit countries. We've seen the gross domestic products of these countries go backwards radically. Life expectancy today is at World War II levels and moving backwards towards the 19th century, not forwards into the 21st.
The most rapidly growing epidemic of HIV is in Russia, Ukraine, and the Baltics. It's estimated by the year 2010 up to 12% of Russians will be infected with HIV.
That is almost a 100% IV drug use, narcotics-driven epidemic. Hey, that's a no-brainer. Make sure that everybody uses clean needles. And start building up methadone programs and drug rehab programs to get the kids off the narcotics in the first place. Guess what? They won't do it. There's a greater hatred of the drug user than there is of the virus.
In sub-Saharan Africa, heterosexual transmission and the low status of women is at the core of the epidemic. It is the only region of the world where more women are infected and dying of AIDS than men.
Number one driving force for this epidemic in sub-Saharan Africa and increasingly in Asia is the extraordinarily low power balance on the side of females. Most women have no right of refusal of sexual intercourse. They have no ability to dictate when, where, with whom. Rape is so commonplace that it's only recently that it's actually thought of as a criminal activity. And men quite commonly have well over 10 women as regulars in their lives.
Garrett says because of the social and cultural issues involved in these countries, AIDS becomes even more complicated to control.
It's one thing to say to a drug user, here's a sterile syringe, at least don't pass your virus to any other drug user. It's another to try and say to an entire society, change all the sexual mores that have been the cultural norm for hundreds of years and become a new kind of society with an entirely different sense of gender balance, of empowerment of women.
In the country of Uganda, however, things are slowly beginning to change. That's because people are talking about AIDS, and there's an aggressive educational campaign to prevent the disease.
There's another factor that's played a big role in Uganda. And that has been the rise of a sense of anger among the women and demanding accountability from their husbands, from the men they have sexual relations with, trying to teach their sons to behave differently.
Today, scientists are recognizing the need for preventive therapies that women can control, such as topical microbicides in gel or capsule form.
The strategy that we've taken is to try to develop a very, very specific inhibitor of HIV that could be applied topically at the site of transmission. So for heterosexual transmission, that would be in the vaginal tract. And that's a very important development that's going on now. And there are companies that are producing these proteins for this purpose. And the hope is that this could be delivered in a practical way all across the world.
DNA vaccines may also prove practical in the developing world.
DNA is fairly stable and may be able to withstand long periods without refrigeration, and that's of course very important in tropical environments and especially in poor places where refrigeration might be a problem.
Scientists agree that public health efforts to prevent AIDS must remain the frontline defense.
We must not lose sight of the fact that we need to prevent it through education, distribution of condoms, needle exchanges, and of course a vaccine.
Many researchers say that the AIDS epidemic is just beginning. While drug therapy has greatly reduced the death rate in the U.S., many countries in the world are facing accelerating rates of infection and death. But research is revealing new ways to fight AIDS, and scientists are beginning to understand the disease not just biologically, but politically, economically, and culturally. A global view that may offer new avenues of hope.
You can't look at the world and see what's happening with 15,000 new infections every day and 8,000 deaths every day, that we can just sit back and be oblivious and not respond.
The real hope lies with taking the younger generation of the planet today, all the teenagers on Earth and the preteens, and as dreadful and horrifying as it may be, getting them to actually imagine this epidemic out of the box, ahead a decade, 2 decades, to the point where they feel motivated not only to protect themselves, but to protect their societies.
All right, so what did we just see in this video? We basically saw a couple of really, really cool instances where people seem to either lack the ability to be infected by HIV or can be infected but never develop AIDS. All right, so in the case of those that can be infected with HIV but never develop AIDS, we saw the story of Eric and that CAF, the antiviral factor that they're looking for. So it seems like there is some sort of immune stimulating factor being produced in people like Eric that keep the virus at low levels, preventing the virus from ever transitioning from the HIV phase into the symptomatic AIDS phase. So that was one case. We also saw a second case involving receptors, where we found people that were exposed repeatedly to HIV, but somehow never become infected. All right, how is this? Well, let's look at the attachment. In early infection, HIV actually attaches to and invades macrophages. This is required for HIV to enter into and establish the initial asymptomatic phase. This is the dormant phase. All right, so HIV must attach to and invade macrophages initially. Then, later on, as HIV makes the transition to AIDS and the symptomatic phase, what happens is HIV then attaches to and invades helper T cells. That is required for the symptomatic phase to begin developing, because it is when helper T cells are infected that the immune system starts to decline and wane and you see the symptoms of AIDS. All right, why are people resistant to HIV infection despite repeated exposure? They lack the receptor on the macrophage that is required for that initial infection. Let's look. This is a figure from the worksheet that I'm going to actually just go to now and show you all, because I've posted this worksheet, emailing it out to you along with this video. And this is what I want you to work through. I'm not going to ask you to print this and turn it in like I usually do. Actually, usually we do this in class and I take it up. But instead, what I want you to do is work through it. Have your answers ready, because when we come in, the date that this is due, and I'm going to let you know that in the email, I'm going to actually do clicker questions to collect your answers on this and to give you points, okay? All right, so what I'm showing you up here at the beginning on this first page is a couple of different things. This is showing you the virus here. It's circular in this image, and you see these little purple blobs. These are the virus receptors. This is the surface of the host cell. You see on the host cell a CD4 receptor and a CCR5 co-receptor. When both are present, the receptor, it binds, and this mediates attachment. All right, this type of attachment would be found on macrophage and be important in the initial asymptomatic infection. Over here, this is actually showing you a different set of receptors. This is actually a T-tropic virus that infects T cells. This is the type of attachment that must occur when the virus is actually transitioning to the AIDS or symptomatic phase, because that's when the virus must be able to attach to and invade T cells. The receptors on the surface of the T cell required is CD4 and this CXCR4. This is the co-receptor on T cells. Both of these must bind to the receptor on the virus for this virus to invade the T cell. All right, what does this have to do with anything? All right, in a recent paper, and I say recent, it's actually from 1996, so probably not too recent, but Paxton et al. Found a long time ago through an infection experiment that people that are exposed but remain uninfected, they actually lack this CCR5 receptor. All right, how do we know? What they did was they took, they had viruses. They had HIV viruses that were M-tropic, those that preferred or that were binding to the CD4 and CCR5 receptor. They had two isolates of that, and that's what you see in these two graphs. And they also had viruses that were T-tropic. They had two isolates that were T-tropic and that preferred binding to CD4 and CXCR4 for the symptomatic phase. So we have early viruses and symptomatic later in infection viruses. And what we did was, they did, Paxton et al., they took CD4 cells and they took, they took T cells and they took macrophage from individuals that were exposed and uninfected, and they mixed them in a tube with the viruses, all right, and compared those to people, control people. So what you see in these images, let's just look. In the blue lines there, the blue squares and circles, this is control individuals. These people are not resistant to HIV infection. And then you see in the orange, these are immune cells that were isolated from the exposed, uninfected. We're calling those EU individuals. If you mix the T-tropic virus with those, and if we measure the amount of virus produced, you see that when we, the M-tropic virus, when we mix the M-tropic virus, either isolate with cells from EU individuals, you see no virus is produced. The virus number, even past days, all the way up to 10 days past exposure, no virus is produced. It remains at 0. However, if we mix these viruses with control individuals, immune cells from people that are not resistant to HIV infection, you see that yes, the virus attaches, it enters the cells, and new viruses are produced, all the way up to 10 to the third. And in this case, a little bit more. higher virus production. So for something is going on, these viruses are not able to infect the macrophage. All right. If we look at the T-tropic virus and we mix those with control individuals and EU individuals, we see in all cases, yeah, the virus can attach and the virus is reproduced by the host cells. Lots of virus. It's going all the way up to 10 to the 4 and possibly more. I don't know how far this scale goes. So what you see in these exposed but uninfected individuals is that their macrophage, which is required for initial infection, cannot be infected. All right. Now, let's look at these following hypotheses about HIV infection. I want you to circle true or false based on the data. Hypothesis 1. Cells from EU individuals cannot be infected at all by HIV. You see low numbers here, but high numbers there. What do you think? All right, number 2. Cells from the EU individuals do not support HIV replication. No virus replication here, but we do see viral replication there. So let's answer that. What do you think? All right, hypothesis 3. M-trophic HIV isolates can infect and replicate in EU cells, but T-trophic cannot. Let's look. Here's the M-trophic. They stay low, but T-trophic cannot. T-trophic goes high. We do see virus. All right, and then the reverse for hypothesis 4. T-trophic HIV isolates can infect and replicate in EU cells, but M-trophic cannot. Answer true or false. All right. Now, please work through this one on your own. I don't want to read this one out loud. I feel like the true or false right above that we just kind of worked through tells you what's going on. It's basically asking you here, M-trophic HIV infects which cells? Macrophage or T cells, so circle 1, and predominates early or late in infection, circle 1, and it uses CD4 and circle 1 of the co-receptors that it uses. And then let's do the reverse, and let's say what T-trophic infects, predominates early or late, and uses what co-receptor. All right, and then let's circle the correct conclusion. What is missing in the EU cells? Is it CD4 missing? Is it CXCR4? Or is it CCR5? All right, where did this possibly come from? This is just an interesting little blurb that I want to add to this. It turns out that looking further into the CCR5 allele, we see that those that are defective in this receptor, they actually have a nucleotide deletion. So we call it CCR5 delta 32. There's actually 32 nucleotide bases missing from that gene. It does result in an abnormal protein that's degraded by the cell, and it never actually makes it up to the immune cell surface. This is why CCR5 is lacking on these people with this mutation. Interestingly as well, we don't seem to see any kind of, side effect or symptom or characteristic from that other than the fact that awesomely they are resistant to HIV. It's not obvious then, how common the allele is, why it came about, etc. So people wanted to look, and they wanted to look around the globe and see, what is the frequency of this mutation? They found that it varied greatly, anywhere from 0 to 14%. Okay, so this is the frequency of the allele in the gene pool, not EU individuals in the population, okay, because everybody has 2 copies of the gene, and so they could be homozygous, heterozygous, you get the idea, right? Anyway, the deletion exists anywhere from 0 to 14% of people. And so what they did was they wanted to kind of make a map and say, well, let's just look at, for example, the varying levels of the frequency. Something really, really interesting kind of panned out from that. We saw, for example, in areas like, tropical climates like Africa here, if they look there, they see, relatively 0. There's no predominance in the population, really. There's hardly any existence at all of this deletion in the gene pool. Whereas if you look at a map of Europe, we see high instances in areas specifically affected by bubonic plague and smallpox. All right, so what's going on? There is a theory that there was a strong selective pressure sometime 700 or more years ago. And we have noted that CCR5, this deletion, gives resistance to HIV, but also gives resistance to bubonic plague and smallpox. So is there something special about this allele and the reason that it's in high percentages in these populations who have basically descended from populations that were highly affected by bubonic plague and smallpox? Possibly. All right, that's all I have for you all today. In this video, what I would love for you guys to do is make sure you are documenting your answers to these questions on this worksheet. Again, I'm not going to make you turn this in when you come back to class, but I will have us actually work through this by clicker, and I will record points for you by that. What I plan to do, just in words, and I'll type this up as well, my plan is for us on January 31st, that would be tomorrow, for us to still continue with our prion discussion. We will work through our prion paper summaries, and we will discuss what prions are and what these papers are finding. And then on February 5th, we will start fungi, protists, and helminths. And February 7th is when I will have you, we will still continue with fungi, protists, and helminths on February 7th, but I'll start out the first few minutes of class with clicker questions to record your answers to this. So these questions should be, you should be ready to click in with the answers to these questions on February 7th. All right? All right, thank you.